Part 1 of Chapter 4 will go through metabolism, enzymes, and how food molecules are broken down to make energy, which is called cellular respiration. The sum of the chemical reactions that occur in our body cells. Anabolism, catabolism are the two types. Anabolism means building larger molecules by putting together small molecules. So here, know that anabolism requires energy. Catabolism, the other type of metabolism, is then to break apart molecules into smaller ones. And catabolism releases energy. A little bit more about anabolism. So anabolism is important for the growth of our body, so multiplication of cells and repair. It does require ATP though. Know that dehydration synthesis, this is just a type of metabolism. Type of anabolism to be more specific. Here, because it's anabolism, this also puts together small molecules to produce larger ones. Know that during dehydration synthesis, water is produced. And so here, our body cells can put together sugar molecules or put together amino acids to make proteins or make fats. In the picture, this is showing how dehydration synthesis works. Here are two monosaccharides, so think these are just sugar molecules. So for them to attach to each other, OH of one molecule and the H of the other molecule, well, they are going to get together, and this is where water comes out. And so then where they used to be, you end up getting this linkage. This O that you see here, this is the oxygen that was not removed from the previous part. Again, so dehydration, here a water molecule is produced. Catabolism is then the opposite. This is when large molecules are broken down into smaller parts and ATP is produced here. Hydrolysis is a type of catabolism. Alright, so here we can break apart sugar molecules into smaller parts, proteins into amino acids, lipids into their components. And so now this is the opposite. This uses water to split the substances. And so it's just the reverse of dehydration. So now the water molecule is going to go where there's the linkage. OH is going to attach to the one molecule, and the H is going to attach on the oxygen of the other molecule. Well, it's very hard for molecules to just break apart on their own or just to come together on their own. Well, thankfully, we have these special proteins, most are proteins, called enzymes. And so these are catalysts, meaning they're not used up in the process and they speed up these reactions of either building larger molecules or breaking them apart. Okay, so know that they increase or speed up these reactions and they lower the activation energy. Now, activation energy is the energy needed to start the reaction at the beginning. And so here, know that what this lowering the activation energy means that with enzymes, less energy is needed because they're doing the work themselves. And they're not consuming the reaction. You can use them over and over. So once they're done with, working on molecules, then they find other molecules to work with. Now, each enzyme is specific to a particular molecule. Now, substrate is just a word to say, the molecules that the enzyme works on. The shape of an enzyme is very important. Here, the enzymes usually have a shape like this. This inside part is called the active site. This is where the molecules attach. Now, if this shape gets distorted, the enzyme unravels, then it's not going to be able to do its job. The factors that can change this shape are too much heat, radiation, electricity, chemicals, if the pH changes, if it goes too high or too low, and some poisons as well. Also know this term, denaturation. It just means the inactivation of an enzyme or any other protein due to one of the factors that are listed above. And so now the enzyme is not able to do its job. In metabolic pathways, more than one enzyme may be used. For example, if a molecule is very large, multiple enzymes may be used to break it apart into smaller parts. Names of enzymes often end in or often contain the name of the substrate that they're working with and end in ASE. So for example, lactase enzyme breaks down lactose. And so here the substrate or the molecule's name is in the enzyme's name. And it ends in ASE. Proteases break down proteins, lipases break down lipids. And so each enzyme works with a particular type of molecule or molecules. Cofactors and coenzymes, these are helpers of enzymes. Cofactors, these are non-protein substances that combine with an enzyme to activate it. Now that's what this picture is showing here. The bottom part is an inactive protein, or think inactive enzyme. Here the cofactor has to sit on it, then the molecules can attach. Without the cofactor, the enzyme or the protein is not able to do its job. And so here cofactors can be ions, elements, small organic molecules. Ion, for example, in hemoglobin, iron atom is needed so that oxygen can attach to hemoglobin. Without iron, hemoglobin cannot do its job. Then coenzymes, these are also helpers. These are organic molecules, most are vitamins. Energy gives us the ability to do work, to change something. Different forms of energy are heat, light, sound, electrical energy, mechanical energy, and chemical energy. Chemical energy, for example, found in food molecules or in wood, coal. But when we burn them, we get energy out. Know that energy cannot be created or destroyed, but we just change it from one form to another. For example, the chemical energy, which is in food molecules, we use that to make ATP. So we're going to go over that, how food molecules are broken down to give us energy, and that's cellular respiration. Before we go into cellular respiration, making ATP, well, what is ATP? ATP is this molecule, short for adenosine triphosphate. It carries energy in a form the cell can use. And so here, food molecules, the energy from food molecules are going to be used to make ATP. So now our cells use this ATP to do the different types of work inside. For example, muscle cells use ATP so that they can contract. So then ATP is going to end up losing a phosphate, we're going to see. ATP has an adenine part, ribose sugar, and 3 phosphates. If we look at this picture here, we can see the adenine, ribose sugar, and 3 phosphate groups. It's these linkages between the phosphates that we're going to see the energy come out. Here, when the cells use ATP to do different kind of stuff, do different work, they're hydrolyzing the ATP. Know what this means. Hydrolysis of ATP just means breaking the bond between phosphates releases energy. When ATP is broken down, imagine this is ATP. When this linkage is broken between the second and the third phosphates, well, now this third phosphate ends up leaving. When that happens, this is not called ATP anymore, but it's called ADP. And so ADP has the adenosine part and only 2 phosphates. And so that's what this first bullet point is talking about. When ATP loses the terminal phosphates, so when we use it, get the energy out, it turns to ADP. So ADP, think this is the used up version of ATP. It has much less energy than ATP. Well, the good thing is this doesn't go to waste. So now, during cellular respiration, when we break apart the food molecules, we turn this ADP back to ATP. And that's how more ATP is made. So here, ADP can be converted back into ATP by giving it, attaching a third phosphate. It's called phosphorylation. And so this is what happens during cellular respiration. And so we're going to go through the steps of turning these ADPs back to ATPs by using the food molecules. When food molecules are broken down, the energy that's held inside the bonds of the food molecules, that energy gets transferred to ATP, but it does also lead to heat. Don't worry about these numbers, but it's saying here at the bottom, 40% of the energy gets converted to energy, but then 60% is released as heat. But hey, that does give us body heat. Okay, so now the major steps of cellular respiration. First step, glycolysis. Notice next to it says anaerobic. This means that during this first step, no oxygen is used. But the next major steps, citric acid cycle and the electron transport chain, those do need oxygen. Therefore, they are aerobic. But yeah, so then we'll talk about citric acid cycle and the last step, electron transport chain, also called oxidative phosphorylation, which makes the most number of ATPs. Well, so these two steps, they do need oxygen. Okay, so we'll look at these different steps. First though, a little bit of an overview for cellular respiration. Know the cellular respiration of glucose. We're going to focus on glucose because our cells love glucose to make ATP. Bye. Know that cellular respiration requires glucose and O2. Okay, so when we break apart food molecules, for example glucose, then what do we get? Carbon dioxide, a little bit of water. We make ATP, but again here the ADPs are getting turned to ATPs, and we do get heat. At the bottom are those two terms again. Anaerobic, if you see this word, it means oxygen is not being used. It's not required. But unfortunately, if you don't use oxygen, you're not going to make much ATP. But aerobic, this means, yep, oxygen is used. Okay, we'll start with the first step for turning those ADPs back to ATPs. First step is glycolysis. So again, it just says first reaction sequence, meaning the first step. Know that here no oxygen is used. In this first step, glucose gets broken down into two. And so here, this first step is only for breaking apart glucose. For other food molecules, the first step is not used, but the other steps, citric acid cycle, electron transport, are used instead. This occurs in the cytosol, meaning just in the liquid part of the cell, so not in any organelle. This is anaerobic, no oxygen is used. And here, only 2 ATPs are made. What's more important, and I'll show you how this is happening in the next picture, but know what is produced at the end of each step. So here, we can put many, many stars at this bottom part. We're going to see that this first step makes NADH. Okay, we'll find out that this NADH is really vitamin B that takes away hydrogens from glucose. Right, so NADH is made, ATP is made, it's just going to be two ATPs, and 2 molecules of pyruvic acid. Pyruvic acid is also called pyruvate. These 2 pyruvates are really just the 2 pieces of glucose, because here glucose gets split into 2 in this step. Okay, so how does this work? At the top is glucose, Now, at the beginning, in order to split up glucose, some ATP does need to get put in. So it is using 2 ATPs at the beginning. This puts phosphates at the 2 ends of glucose. So now glucose splits up. Now this is where vitamin B comes in. NAD+. This is actually vitamin B3. This is able to take away, it takes away hydrogens from the piece of glucose. And so when it takes away the hydrogens, it turns to NADH. We're going to see this NADH get used to make a bunch of ATPs at the very last step, which is the electron transport chain. Okay, then we can start to make ATP. So here, ADPs are going to pick up those phosphates. So when they pick up the phosphates, they turn to ATP. So again, as a summary, what is being produced at the end of glycolysis? This makes NADH. We get only 2 ATP and 2 pyruvates, which are the 2 pieces of glucose. Again, pyruvates are also called pyruvic acid. If there is oxygen, then pyruvic acid or the pyruvates go into the next steps. There's an intermediate step of when pyruvate turns to acetyl-CoA. I'll show you on the next picture. But then the next major step is going to be the citric acid cycle. And then the last one is the electron transport chain. And so in the intermediate step, pyruvic acid is used to produce acetyl-CoA. So here, acetyl-CoA is getting produced. Pyruvic acid enters the mitochondria. Here again, NADH gets produced. And then acetic acid is going to combine with acetyl-CoA, coenzyme A, to become acetyl-CoA. But here, more importantly, so now after that comes the next major step, citric acid cycle. In the citric acid cycle, now this molecule is going to go through some changes. Here, NAD, that vitamin B3 comes in again, takes away the hydrogens, turns to NADH. ATP gets produced. Carbon dioxide is also getting produced here because the carbons and the oxygens from the piece of glucose are being taken away. And FADH is produced. Now, FADH2, before it takes away the hydrogens, it's called FAD. Now, this one is actually vitamin B2. Alright, so the important stuff is what gets produced during the citric acid cycle. Now, the citric acid cycle, it does run twice because it's running one time for each piece of glucose. Well, we have 2 pyruvates, therefore it's going to run twice. And so total, though, for the 2 pieces of glucose, citric acid cycle is going to make 2 ATP. It's going to make NADHs, FADH2, and CO2. Now we can go on to the last step, which is going to make the most ATPs. Now we're going to use up the NADHs and the FADH2s. So now these are going to give away those hydrogens that they took away from glucose. So here they're carrying the hydrogens and the electrons of the hydrogens to this ETC, which is the electron transport chain. And it's going to occur in the mitochondria. Now let's look at how this is happening. Okay, so this is showing the last step, electron transport chain, also called oxidative phosphorylation. The mitochondrion has 2 layers of coverings. And so here we can see the picture of the mitochondrion. There's the outermost covering, but then kind of in the inner part, there's a lining, and it ends up making these weird extensions going into the cell. And so this is what we're seeing here. In the upper part, This is the outermost membrane, but then this part is the inner covering. Now in this inner covering, this inner membrane, are these special channels. And so this is where NADH and FADH2 come over and they give away their hydrogens. So now their hydrogens are being moved to this intermembrane space, meaning the space between the outer membrane and the inner membrane. And the electrons of the hydrogens are being passed along these proteins. Okay, so now we have a bunch of hydrogens in this intermembrane space. So I'm going to write it on this side. We have a bunch of hydrogens on this side, but not so much on the inner part. Well, remember diffusion. In this case, it's facilitated diffusion. So here, these hydrogens are going to end up going through this last channel. Now, this one is special. This allows the hydrogens to go back down instead. The previous ones made the hydrogens go up into the inner membrane space, but now this one allows it to go high to low. And this last channel is called ATP synthase. All right, so when the hydrogens go through it, it ends up powering it, this ATP synthase. It ends up turning like a turbine. So when it does that, it actually acts as an enzyme. It puts together ADPs and phosphate pieces together to make ATP. All right, so know that during this last step, this ATP synthase channel is used, which makes ATPs when the hydrogens go through, high to low. But, so this only works if the hydrogens go high to low. But what would happen if the hydrogens ended up collecting here at the bottom? Then you wouldn't have this high to low difference. We need something to take away the hydrogens so that they don't collect here. And what is going to do that is oxygen. Oxygen grabs these hydrogens after they come through. It grabs the electrons of the hydrogens as well, and we get H2O. Alright, so this oxygen is absolutely necessary for this to work, because then you're not going to have this high to low hydrogen difference anymore. We need oxygen to take away those hydrogens. This picture is showing the ATP synthase. Again, here you go from high hydrogen to low hydrogen. Alright, so we can hear it star ATP synthase. Note that this is used during this last step, electron transport chain. Again, this step makes the most ATP. Now, the number of ATPs can vary. It depends on if things are going well according to plan, but here we're going to end up making 24 ATPs. Oh, wanted to point out, if you see the word proton, this just refers to the hydrogens. Okay, so they move through the channel ATP synthase. It only works high to low. Okay, like I was saying before, oxygen is absolutely necessary for this to work. If there is an oxygen around in the absence of oxygen, then there is no acceptor. So here, what happens to the pyruvates if they're not able to go into the mitochondria to do these next steps? After glycolysis, pyruvates were made. What happens is those hydrogens that are picked up are then given to pyruvic acid. So the pyruvic acid, or the pyruvates, turn into lactic acid. And so here I'm going to put lactic acid forms if there is low oxygen, and it forms when pyruvates pick up those hydrogens instead. And so in the absence of oxygen, only the first step occurs. And so glycolysis is the only step that occurs. Hey, it's still making ATP, but we're making much less ATP than if we did the other steps as well, if there was oxygen. Okay, so then it ends up giving us a net gain of 2 ATPs per molecule of glucose.